Morning, everyone. My name is Javad Rahimikollu. I'm a PhD candidate in Joint CMU with Computational Biology. I work in systems immunology department at University of Pittsburgh under supervision of Dr. Jishnu Das. Today, I'm going to present multidimensional integration of PPI network with genetic and molecular data to decipher genetic underpinnings of RA endotypes. So before our presentation, our lab have 3 oral presentation in different sessions. I am presenting in TransMed. My colleague and my lab mate, Jane Syvick, is going to present a very interesting talk in a computational system immunology session called SWING. And my advisor, Dr. Jishnu Das, in ML-CSP today is going to present a slide. It's a very good work published in Nature Methods. Please attend the talks, and we would love to hear your feedbacks. So, rheumatoid arthritis is a progressive autoimmune disease associated with substantial molecular and cellular heterogeneity. Clinical detection of RA is based on presence of anti-CCP and RF factor markers in the bloodstream of the patients. In our study, we got roughly 1,000 subjects with clinically diagnosed rheumatoid arthritis from the RACER cohort with corresponding clinical metadata and individual genotype data. Here, I would like to thank our collaborators, Dr. Zhang and Dr. Moorhead, with their amazing insight and guidance and helping us pre-processing our data. In this study, we had roughly 5 patients who were double seropositive, and roughly 400 patients were single positive. Despite the difference in the progression of the outcomes, the subjects get same treatment regimens. So the key question is, do these 2 disease subtypes correspond to distinct disease states with a difference in genetic component and pathophysiological basis? Are they 2 disease endotypes? Are there basically different disease presentations? The goal of this study to develop a framework to integrate genetic, molecular, and cellular data to study and examine this heterogeneity between doubly seropositive and single positive RA. So I'm going to walk through this framework step by step through the talk. The very first question of this study and the framework is that, is there any difference in the genetic components of 2 disease subgroups? So we need to have a measure for this genetic difference. We use heritability to measure the genetic difference between these 2 disease subgroups. Heritability is the proportion of the variation in the phenotype explained by the genotype. And as we see that heritability of the difference in the heritability between these 2 subgroups is 30%, while the heritability of RA versus control is 60%. This is almost half of the heritability of the entire RA. The result is significant, and our conclusion is we are dealing with an endotype. So if we have a genetic signal, we had this difference in the heritability, and we want to know what part of the genome is driving this signal. Unsurprisingly, as we expect, the signal is coming from the HLA region of the chromosome 6. So we are dealing with an autoimmune disease. This is not a surprise. We do a zooming plot when we see that the signal is actually coming from HLA-DRB5 gene. Surprisingly, we see that the heritability of this region is 3%. We already said that the heritability that we calculated from the whole genome is 30%. This is much less than the heritability that we expected. So we are dealing with a missing heritability problem. So can we identify an additional loci that are more complete in explaining the difference heritability between double seropositive and single seropositive RA? So since we had a missing heritability problem with the GWAS, we decided to take an alternate approach. One of the main drawbacks of the GWAS that it assumes that the loci are independent from each other with no biological relationship between the loci. Network GWAS, on the other hand, tries to take into account the gene relationship among the loci using biological networks. There are multiple studies already shows the advantage and more accuracy of network GWAS compared to traditional GWAS methods. In our GWAS approach, we are using network propagation to integrate experimentally validated PPI network with our genetic study. Here we are using HotNet2 to do the network propagation. HotNet2 is a heat diffusion method that spread the heat of the nodes throughout the network using a technique called random walk with restart. The goal of the propagation is to find high scoring subnetworks and modules in the network that are relevant to our endotype of interest. To find heat of each network, we use this method called linkage disequilibrium adjusted kinship method. So, as already I explained, we want to find high significant, high scoring communities in experimentally validated PPI networks. And as you see, the search space is very large. However, despite this large search space, we were able to identify a very specific modules from this big network. After finding these network modules, the next step is to investigate whether these modules are biologically meaningful. And for that, we have 3 different sets of validation. The first validation is on the genetic data. It is the data that we trained our networks. The goal is to determine which identified modules have a significant contribution to difference in the heritability that explain the difference in heritability of 2 subgroups of doubly seropositive and single positive RA. We see that 4 out of 14 modules are statistically significant. Moreover, we are able to mitigate the missing heritability problem. As an example, the HLA region in GWAS had the heritability of 3%, but however, module 1, which is our HLA region, has a heritability of 7.5%. And this is an indication of increasing power in our genetic study. Next, we are going to the second round of validation. In second round of validation, we use genotype to impute expression data for each tissue. For that one, we are using PredictXscan method. The goal is to see whether the genes from the modules that we identified using the genotype and the protein-protein interaction are discriminatory between double seropositive and single seropositive in the imputed gene expression data. So these are the results, and these are also very interesting because these results not only give us the modules that are significant in discriminating these 2 groups, but also it gives us tissue specificity that which modules are significant in which tissue. So far, we have validated modules using genotype and imputed expression data. Next, we are going to the next step. We try to see if whether the genes of module are discriminatory in an orthogonal bark RNA sequencing data. So we got all bark RNA sequencing data from our collaborator, Dr. Zhang, which includes sorted bark RNA-seq data for different cell types of T cell, B cell, fibroblasts, and monocytes. The results lends cell type specificity and also identifies the significant modules. This validation is very significant. Since the modules are trained in genetic and protein-protein interaction data, first of all, this is a different modality. This is gene expression, and this is from a complete orthogonal study. In summary, we captured 4 different modules, including known HLA region by integrating PPI network to our GWAS study. We also find out that these 2 groups of RA are endotypes with different genetic bases. We not only find the significant modules, but also we find the specific tissue and the specific cell types that these modules are enriched, and which cell types that are actually relevant to our RA endotype. I would like to thank my advisor, Dr. Jishnu Das, for his amazing and excellent guidance throughout this project, and Priyambada Gohar Roy, who has led this project with me. And I would like to thank my lab members, amazing lab members in DAS Lab for their interesting support. With that, I'm happy to take some questions. Thank you.
Wonderful. Thank you so much. We have time for one quick question.
Thank you very much. I was wondering which type of network you used and if you tried with different network types as a background for HotNet.
Yes, we use protein-protein interaction network. That is context agnostic. We could have used other networks like co-expression or GRN networks. For that you need to have data, and those actually depend on the context. We got our network from Hint. All of the protein-protein interactions that we use are experimentally validated. And if you want, I can share you the website and the database that we use.
Fantastic.